All right, well, I've got 7 o'clock. Patricia, you might just introduce yourself. There may be some people who've never met you, and then go ahead and get started. As you can see, obviously, today's topic, asthma and the microbiome. Go right ahead.
Great, thank you. For those who don't know, my name is Patricia dela Cruz. I'm one of the first year allergy immunology fellows. And as you can see, I'm going to be talking to you all about asthma and the microbiome. To no surprise, I have no disclosures. And so before I get into the meat of asthma and the microbiome, I first wanted to start off with a general introduction about the microbiome itself and how it could potentially impact health and disease. And so here you can see on the left is the cover of Nature, previously where titled Fellow Travelers, where the first results from the Human Microbiome Project were published and really highlighted that healthy variation of quote unquote our microbial selves. And sort of describing this as our microbial selves is appropriate because by the numbers you can see that there's 10 to 100 trillion microorganisms, about 2 to 5 pounds in weight. And sorry, you all, my screen looks a little odd for my view. Does everything look okay to everyone?
Yeah, your slides look fine.
Okay, great. It's just my screen. All right, no problem. And there's over 1,000 bacterial species, 3.3 million unique genes in the gut. Actually, the ratio, we're outnumbered. The ratio of bacteria to human cells is 1.3 to 1. So showing this photo of quote unquote our microbial cells is quite appropriate in this regard because there's this huge community of microorganisms living within us, having this symbiotic relationship with us. And so looking at the composition of the microbiome, it actually varies at different sites of the body. Of course, much of the focus initially was in the gut, where a huge burden of that microbiota reside. But as more studies were done, previous sites of the body that were thought to be sterile, or more sterile rather, such as the respiratory tract, were found to actually have a microbiota and microbiome in itself, where there's resident microorganisms there that help interact with the epithelium and with immune cells there to help impact health and disease. And for the purposes of this talk, I'm of course going to focus more on the gut and the respiratory tract. So with this slide, I just wanted to emphasize the concept of sort of balance and interplay. And it's not just a matter of just good bacteria giving good bacteria or too much of bad bacteria, things like that. But it's really an interplay with the host and the microbiome. So you can see on the left is a healthy gut microbiota, where of course the bacteria are primarily in the gut lumen. They are kept sort of at bay in this symbiotic relationship with the host, and there's actually crosstalk between the epithelium and the immune cells and the bacteria that reside there, where there's this sort of physiologic amount of inflammation in a healthy environment. However, when there's an altered gut environment, perhaps externally, for example, in the context of antibiotics, diet, hygiene, pollutants, virus, there's this state of dysbiosis where it's imbalance. And you can get damage of the epithelium, as you can see here, uh, increased bacterial adherence and penetration, actually translocation, causing this pathologic inflammation. I say all this with a caveat that while it could be altered with external stimuli, certainly genetics and intrinsic factors are at play as well. For us as fellows and for others on the call who see folks with primary immunodeficiencies or inborn areas of immunity, there's such pronounced intestinal manifestations in certain severe monogenic immunodeficiencies and certain immune gene variants. And so it's going to be an interaction between genetics, the environment, and the microbiota that is there that will sort of help either promote this balance or have this a dysbiosis. So there's multiple mechanisms by which the microbiome can impact the body. You can see here on the left is a state of homeostasis. There's microbial products, CpG, LPS, things like that, that can interact with, again, the epithelium and immune cells, metabolites such as short-chain fatty acids that can be both either activating, inhibitory. There's physical translocation of bacteria in states of inflammation. And in all of these cases can have local responses and local impact on the body, but can also impact more broadly, getting mediators such as cytokines like IL-17 or IL-22 into the circulation. And in terms of translocation of bacteria, both translocation of bacteria and bacterial products can certainly activate receptors more broadly if they get into the circulation and impact the body in that way. I list here enteric nerves just because there's also a growing amount of research focusing on how the impact of the microbiome and the immune cells within the intestine can actually affect the rest of the body through the enteric nervous system. But with my talk being really about asthma and the microbiome, of course, I'm really discussing this gut-lung axis where there's this state of eubiosis, healthy lung microbiome, healthy intestinal microbiome, good barrier integrity. And then there's also states of dysbiosis. There's actually mechanisms by which there can be gut-lung crosstalk through immune cell mishoming, mediators from the immune system, going to circulation, going to the lung, breaches in the epithelial barrier, et cetera. Later in the talk, I'm also going to talk about the gut microbiota restoration with pre or postbiotics, pre or pro biotics, but a bit of a spoiler alert, certainly more mechanistic studies need to be done to be able to optimize and guide therapies in that way. So with that, I'm going to go over a few studies that have shaped what we know currently about asthma and the microbiome. A lot of this is in the earlier states in descriptive studies, and I'll focus all on human studies today. But a lot of these are descriptive and associations, and I think the field is certainly moving with the tools that it has in terms of looking more at causation and to be able to develop therapies in that regard. But as you'll see, a lot of this is more observational studies. And then for anyone here that's familiar with these studies or has more expertise, feel free to chime in. It's certainly an area that I was really learning about in the creation of this talk. And so I'll start with the sort of hygiene hypothesis, of course, with the idea that this exposure to environmental microorganisms can impact the risk of development of atopy and allergic disease, including asthma, which sort of lends itself well to what intrinsically is happening in terms of the microbiome. What does that look like in patients with asthma, both in the intestine and the respiratory tract? I'll talk about some associations with clinical data and then some preliminary clinical interventional studies that have been done. And so I wanted to start with this paper from 2011, which is hard to believe that's almost 15 years ago. But it's the exposure to environmental microorganisms in childhood asthma that was done in the New England Journal of Medicine. And in this study, they looked at these two cohorts, the Parsifal cohort and the Gabriella cohort. And what was particularly special about this cohort is that there was a large proportion of them living on farms. So the Parsifal cohort is actually 52% of the children living on farms, and the Gabriella 16. And so this gave a unique opportunity that they were able to analyze the environmental microorganism exposure to those living on farms compared with age-matched, population-matched controls within the respective cohorts themselves. In addition to looking at development of asthma, they of course looked at the environmental microorganisms, and they did this primarily through looking at dust analysis and microbial analysis on that collected from the environment, like the mattress, vacuum, things like that. What they saw was that in both cohorts, the Parsifal and the Gabriella cohort, that the children that were living on farms here in this sort of brown pink color had a decreased prevalence of asthma compared to the reference group, and similarly atopy. When they looked at potentially why in that microbial analysis, they found that diversity matters. In this case, of course, bacterial diversity matters. So this graph here, you can think of it as sort of looking at two separate graphs where sort of the blue is one and the red is the other. So if you look at the y-axis, the probability of living on a farm, which is going to be your y-axis, is significantly associated with an increasing number of detectable bands, which is just a surrogate marker of their bacterial diversity with their assays. So that has a positive correlation and very clearly an inverse correlation associated with the probability of these participants developing asthma, suggesting that bacterial diversity is protective potentially in some regard. But you can see the statistics down here. I focus mainly on the Parsifal cohort just because of the particular bacterial studies that they were able to do. But the unadjusted odds ratio is 0.62 with a confidence interval listed there. And interestingly, even if you adjusted for living on a farm or not, the diversity of the environmental microorganism specimens were still remained significantly and positively associated, inversely associated with the development of asthma with a significant p-value. And so these studies really looking at the environmental microorganisms lent itself well to the microbiome studies that I'm going to go into, because then the questions were, that's what's happening from environmental microorganisms, what's happening with it now that we know that there's this whole community of microorganisms within us, how is that then impacting the development of allergic disease? And as more studies are being done on this, a lot of focus was placed actually on early childhood. And the reason why is because there was this concept of this critical window of time where the infant microbiome and its metabolites were actually associated with immune tone and later development of asthma and allergy. So while I'm going to focus on asthma itself, this has been shown in allergic rhinitis, seeing differentially methylated CpGs in the upper airway, atopic dermatitis, of course, with skin colonization with staph, and again, my focus being the disruption of the microbiota in the context of asthma. And so what does the intestinal microbiome look like in asthma? So one of the studies that I'm going to review is called the COPSAC 2010 birth cohort study. It was done in Eastern Denmark in 2008 and 2010. And so for what it's worth, of course, there's probably a lack of ethnic diversity in that regard, just to call that out. But for the methods of study, they had structured interviews at 11 visits during the first 5 years of life, looking at asthma as the primary outcome. 51% were male, 57% had siblings at home, 22% C-section, and 46% were treated with antibiotics in the first year of life. The mean maternal age was 32, and 26% of the mothers had this physician-diagnosed asthma. And the reason I bring up this patient breakdown is because there's been multiple other studies that have shown that individually, many of these factors, such as C-section birth or antibiotics during the first year of life, can impact the microbiome. And so with this study published in Nature Communications in 2018, they found that in early life, at 1 year old, patients with asthma later have an altered microbiome. And so I know this is a bit complex figure with a lot going on. Just to take it step by step here, the y-axis is the bacteria, the x-axis is the relative abundance, the orange is asthma, and this sort of teal green color is no asthma. So looking at the first panel alone, this is all the study participants in N of 589. While there's certainly similarities amongst the composition of the microbiome, there are significant differences in specific bacteria between those that developed asthma later and those that did not. What I found particularly interesting in this is when they dichotomized the data by patients who were from a mother that had asthma versus no asthma, there was such a striking difference. In this middle panel, these are all the patients that had an asthmatic mother, and there were essentially the significant differences seen when the group was all together were made much more significant, much more dramatic. Whereas these differences that were seen previously are essentially minimized in the non-asthmatic mother group, really suggesting a component of genetics potentially at play, and really harping back to that interplay between genetics and the microbiome, and potentially how critical that may be in sort of this establishment of the microbiome early in life. Also in this study, they looked at how does the microbiome change over time in this sort of critical window. And so they looked at 1 week, 1 month, and 1 year. So these aren't separated by asthma or not. This is just all the participants together. And you can see that looking at this panel A, 1 week is in gold, 1 month is in purple, and then 1 year is in green, that the 1 week and 1 month look closer together. And then there's this shift over time to what the microbiome composition typically looks like at 1 year old. So sort of very clearly visualizing the fact that there's 2 clusters. They did statistical analyses and then separated these into 2 clusters. And you can see very similarly to what I just described, the red or cluster 1 is more typical in this 1 week of life, 1 month. And then there's this quote unquote maturation or development over time where that shifts to more this cluster 2 at 1 year.
Patricia,
And the reason
can
go
you
ahead.
explain this slide a little better? What's being plotted here? I don't understand this picture.
Yeah, sure. So they are essentially clustering the microbiota at 1 week, 1 month, and 1 year. And so it's looking, the way I think about it is that when it's closer together, it's more similar in how the composition of the microbiota looks. And so for the goal, if you look at panel A, for example, the gold and the purple are closer together in this plot and cluster together, and they're more similar in their composition, just as a sort of cursory way of interpreting that. Whereas there are more different at 1 year old, you can see it's sort of shifted in that regard and further away from that clustering. And so what the panel in C is looking at is what, if we separate it and look over time, what does a typical microbiome look like? And what it's really just showing, the take-home message is that at 1 week and 1 month, they're quite similar.
So are these kids with asthma or all comers, or who are
These
they?
are all comers. Yeah, these are all comers. They didn't separate it with asthma and not asthma yet.
And what are in the clusters? Different microorganisms?
Correct. Yeah, the composition of the different yeah, microorganisms.
Okay.
And the reason I'm really going over this is because they looked at how this composition potentially affected risk of asthma at 5 years old. So that was all batched, no difference between asthma and non-asthma. And what they saw was that lack of maturation, so that lack of progression, shifting from this sort of looking like a 1 week, 1 month, and looking to a year old. If you don't look like this sort of matured, quote unquote, matured microbiome at a year old, your risk of asthma is increased. So you can see in this first panel, this is patients from both asthmatic and non-asthmatic mothers with a risk of, if you're at 1 year old, you looked like the red, which is more representative of the 1 week and 1 month, you had an increased risk of persistent asthma. Again, breaking it down by patients who come from mothers with asthma versus mothers that did not have asthma, you can see that much of this effect is substantially increased in patients with asthma, again, highlighting and underlining that genetic component to it. And so that's sort of one study in terms of looking at how the microbiome is different, potentially in patients with asthma, and how it can impact risk down the line. Similarly, there were questions about what about the respiratory microbiome, and I focus a lot on the nasal microbiome just because it's easier to sample, and people don't unless things are going wrong, aren't routinely getting bronched and getting lower airway sampling. And similarly, kids are unable to produce sputum to really get that lower airway sample in the pediatric population, or a surrogate sample. And so with this respiratory microbiome, there was also the question of whether or not there are this sort of critical window in early life development is also important, or if there's changes there. And this is from a review by Shah and Bunyavanich that showed that yes, indeed, when certain bacteria are predominant earlier in life, and the composition looks in the way that is displayed here, then there's an increased risk, or rather an association of development with recurrent wheeze and asthma in childhood in this early life phase. So things like strep pneumo, I'm sorry all if you hear my dog in the background, strep pneumo, H flu, Moraxella, Prevotella, and things like that. I can go into a little bit more detail about some of these studies, but just to highlight that establishment of the microbiome in the airway earlier on, similarly to the intestine, can affect risk of development of asthma down the line. But it's not to say that outside of this early window, there's no further effect, that no other changes that can be done. There's multiple factors that influence the airway microbiome and the development of pediatric asthma. For example, in this state, that includes birth characteristics, diet, the environment, respiratory illnesses, antibiotics, and again, host characteristics, including genetics as well. And so in that regard, in terms of going beyond this sort of critical early window, this study looked from still pediatrics, but age 6 to 17, looked at how the composition of the nasal microbiome could potentially differentially relate to the exacerbation in pediatric patients with asthma. And so this was, again, ages 6 to 17 during the fall season, so August to September through November, December. And they had weekly nasal samples and respiratory illness assessments with a total of 413 participants from which they were able to get these nasal samples. And what they found was that there were distinct bacteria where, when they predominated within the nasal microbiome of these participants, were differentially associated with the risk of various things, including exacerbation within that time frame, that August to November, December timeframe, rhinovirus risk, and respiratory illness risk. And so just taking it one by a step by step here, this group, I'm not sure if you all can see my pointer, but this group of Haemophilus, Aleococcus, Corneobacterium, and Staphylococcus, when those were predominant, they were associated with a decreased exacerbation risk. You can see the relative risk here in this top row, 0.41, 0.6, 1.44, 0.70. Conversely, when Moraxella predominated, which was more prominent in the younger patients, there was an increased exacerbation risk within that window as well. They also highlight the risk of rhinovirus infection, and which sort of lends itself to discussing that interplay between the bacteria and the virome. But rhinovirus is certainly one of the leading causes of pediatric asthma exacerbation. You could see that both Haemophilus and Streptococcus have a dramatically increased causes an increased risk of rhinovirus infection, and decreased with staph, and similarly respiratory illnesses are decreased with these with Corneobacterium and Staphylococcus, and increased with strep. And so they were asking what mechanisms may be at play to cause some of these different changes in risk.
But Patricia,
And so,
1 second before you go ahead.
yeah.
This is different, though. This is not what microbiome has to do with inducing asthma de novo.
Mm-hmm.
This is people who already have asthma, and just what organisms are related to risk of exacerbation once they already have it. Is that right?
Correct. Yeah.
Okay.
So not just in the development, but once you already have it, then what might be causing or contributing or increasing your risk of these things.
Actually, while I've interrupted you, I wanted to go back to the very first study you showed of farm exposure.
Hmm.
Is there an implication that the external organisms that you're exposed to if you live on a farm then become part of your internal microbiome? Is that known, or are those independent ideas?
Yes, I had the same question because in much of the microbiome work they sort of led with this hygiene hypothesis, but I didn't see studies, and it might just be because I'm not familiar with the studies that were done, but I didn't see studies that directly looked at the how that farm exposure either directly contributes to the microbiome of that state, and if that effect is is sustained over time, and whether or not that that sort of changes back. The genetics component in that study really suggests that there's this strong genetic predisposition in that regard. So I think the question is if this indeed becomes part of the microbiome over time, will the genetics sort of allow it to hold up, and how will it affect disease later? I mean, I couldn't find studies on it, but you know, not to say that they don't necessarily
They
exist.
probably haven't been done, but I mean, it did
Hmm.
look from the data you showed, um, sort of combining if the mother had asthma or didn't have asthma. It looked like the genetic effect of of a mother with asthma was greater than the influence of the microbiome.
Yeah,
Yes.
I agree. I mean, I think that they by showing that genetic predisposition, it begs a question because I think it was always, is it the chicken or the egg? Is the microbiome affecting the disease, or is the genetics affecting the microbiome then affecting disease? And I think their argument is that yes, the genetics seem to outweigh um, the the potential effect, but it sort of acts as a setup for what the microbiome looks like in that regard. And from there, the metabolites, things like that, can then potentially play a role.
Okay. Go ahead.
So this next chart is just looking at some of these um, when these certain bacteria predominate in the airway. They looked in vitro in human airway epithelial cells, essentially stimulated them with biofilm derived products of these specific bacteria to start to get a sense of what might be going on immunologically that is impacting disease. And you can see with the Warwickella, there's a significant increase in the gene expression of IL-33 and IL-8. IL-33, of course, implicated in the pathway with IL-33, TSLP, things like that. And IL-8 is also CXCL8, which is a potent neutrophil chemokine. LDH they used as a surrogate for epithelial damage. So they also see that with Moraxella, there's an increase in the amount of LDH or epithelial damage as well, all potentially getting to maybe ways that this particular bacteria is interacting with the host and causing issues in terms of increasing the risk of certain infection, as previously stated. But I think one sort of point that this brings up, where there's differences in the nasal microbiota of patients with asthma versus patients without asthma, and there's differences there. I think this study also brings up the point that within all patients with asthma, there's differences between them as well, getting to the fact that asthma is heterogeneous disease. And so this is a slide just talking about one the concept of this umbrella of asthma and bees. And while we get these observable phenotypes, such as the clinical presentation, symptoms, and observable characteristics, what's really exciting in this line of work is that they're starting to characterize different endotypes of asthma. which is specifically distinct biological mechanisms that links these clinical characteristics with a particular mechanistic and molecular pathway, with the hope that there's some personalized therapy that can be done to specifically address that particular patient's disease. And so there are studies, and it's just taken from a review just to condense it here, that when you dichotomize the asthma between type 2 high or type 2 asthma versus non-type 2, that there's a difference in the airway microbiome as well. Again, you'll keep seeing similar bacterial genus pop up, with Haemophilus being high in T2 low, and Moraxella also being high in T2 low as well, Neisseria and Fusobacterium. With Fusobacterium being high in the T2 high adults. Again, just very descriptive work. Similarly, the part of the different sort of phenotypes that's seen in asthma is the sort of neutrophilic type asthma versus the eosinophilic type versus a polygranulocytic asthma. This was data taken from a cohort that was from Australia, recruited across 8 centers. And these were adults with induced sputum with patients with severe asthma. And they saw that in neutrophilic asthma, here in the blue in this first column, that there is lower bacterial diversity in the sputum, which is their surrogate for the lower respiratory tract, compared to those with eosinophilic asthma or polygranulocytic asthma. And that this diversity was inversely associated with the amount of neutrophils that they found in the sputum. Shifting gears a little bit, a lot of that was descriptive studies about what the microbiome looks like in the context of asthma and potentially exacerbation and things like that. And so I'm shifting a little bit into the metabolome in terms of the bacterial products that are produced and how that could potentially affect asthma. So this next study was from the VDART trial, which is a vitamin D antenatal asthma reduction trial. This was a multi-site randomized double-blind placebo-controlled trial, vitamin D supplementation for prevention of asthma and other allergic disease. I'm not going to focus on the vitamin D part, but rather they were able to collect stool samples and look at the microbiome of this patient cohort and look at the potential development of disease. So it's an integrative analysis, birth to 3 years old. Out of the 806 participants, they were able to have 361 stool samples for metabolomic analysis. And then they tried to see what was associated with diagnosis, physician asthma diagnosis by age 3, and if there were any correlations with the plasma metabolites that were available, and if there was any association with food. So patients were given, and the parents were given, food frequency questionnaire that they filled out over time, and you'll see that they were able to certainly find some associations in that regard. Okay, and so when they looked at this data, they saw that there were certain intestinal metabolites that were associated with childhood asthma at age 3. The way I'm reading this is there's these blue dots here. All the blue dots are significantly associated with asthma, and certain intestinal metabolites, including endocannabinoids, which I sort of looked up what each of these things tend to do in the context of the immune system, and they're involved with the regulation of immunometabolic processes, fatty acids such as omega-3 fatty acids, and then these diacylglycerol metabolites, which are involved again in immune signaling cell growth. It's also used as emulsifiers in different foods, but they found that there are certain metabolites within these categories, specifically the ones in blue, that are inversely associated with the odds on the y-axis, the odds ratio for association with asthma. So the next question was, what potentially could be the source of these intestinal metabolites? Is it diet? Is the microbiota? Is it extrinsic or intrinsic? What they found was that there were specific bacterial families in the sequencing, specifically this particular family that is associated with the intestinal metabolites that they found that could potentially be playing a role in asthma. Interestingly, when they looked at the diet and how it could relate, I looked at the questionnaires and they asked the question, which foods um, are have associations with the intestinal metabolites and with risk of asthma development. And they found essentially many of my favorite foods are associated with specific metabolites that are also associated with um, asthma down the line. So the thought that a lot of the fried or processed meats, fried chicken, hot dogs, hamburgers, things like that, have associated metabolites that again have associations with asthma. And they sort of purposely didn't look at the direct association with, say, fried chicken and asthma, because the goal really was what could potentially be impacting asthma mediated through these specific intestinal metabolites. And finally, they found that some plasma metabolites, such as gamma and beta tocopherol, were associated with these intestinal metabolites as well. There is a growing amount of other studies that I'm not going to really go over that identifies a whole host of different bacterial metabolites that can have an impact on immunity and asthma. And I won't go over all of this, but just to say that there's an increasing amount of research being done and some mechanistic work being done to hopefully start to figure out what of these are targetable and what could potentially be therapeutic.
Last question, go back to that food slide. This one. So is the point here that if these things are in your diet, you're feeding certain microorganisms that then make metabolites that are bad? Is that the bottom line?
Yeah. I mean that's essentially it, that these are potentially
Yeah.
causing microorganisms that produce these metabolites that are associated with asthma.
Don't eat
Yeah.
anything on this list is the point.
I know, except the issue is I ate many of these things just yesterday, and it's such a common thing. And so I think certainly much more research needs to be done than say, don't eat potatoes, which is crazy. Um, and so I think more research needs to be done on certainly what specifically um, causes these associations, and to what degree does it really account um, for the mediation between the relationship between diet and asthma.
Okay.
All right, last bit that I'll just touch on is probiotics and prebiotics, and some of the work that's been done looking at is this a treatment option, either for prevention or for treatment. So a few definitions. One, probiotic, which is live microorganisms that when administered in adequate amounts can confer potentially health benefit to the host. Prebiotic is a substrate that is used by that microorganism that promotes the growth of that microorganism, conferring a health benefit. And then there's other ones in these meta-analyses that I'll show that's called a symbiotic, so it's a mixture of the two. And so I'll pull up sort of two meta-analyses that I saw. One included 12 studies with randomized controlled trials with individuals that were already diagnosed with asthma. And this was an administration of probiotics. I put this here on the right. It's not inclusive of all 12 studies, but I think shows that some of the studies are not well powered, with 4 and 4 in terms of the number of participants, while others are larger in terms of going into the hundreds. So one of the ones that I'm gonna talk about is this one, the Drago et al. In 2022. That's more powered in terms of about 200 per both interventional group and placebo group that were given lactobacillus and bifido for 16 weeks. In this report, they found that there was a decreased exacerbation rate in patients that were given probiotics. This was a randomized controlled trial in Italy, again, about 400 children mean age of 7 years old, and they were given it for 16 weeks. And you can see the odds ratio is 3.17, suggesting that potentially there is this protective effect of probiotics in this cohort. People looked at other clinical parameters like FEV1, saw no effect. FeNO, peripheral EOS, saw no effect. And a whole host of different trials, not just in the United States and California, but in the EU and in other places around the world. And I only put this on here to show, looking at this, the far right-hand column, is that a lot of them show no difference across the board in terms of development of asthma, development of allergic disease, reduction in certain parameters. And I think that in part is because we need more studies to figure out what bacteria might be helpful, or should we really go for bacterial products because of the genetics that are at play, and just more mechanistic studies to be able to guide development of treatments, potentially harnessing and utilizing these pathways, and also to identify who might actually benefit. Just because with the data that's there, it's clear, similar to the fact that there is heterogeneity amongst the mechanisms that play in asthma and the different endotypes, and that not all biologics are going to be effective for certain types of asthma. I think that concept can be similarly applied in the manipulation of the microbiome and bacterial products, that there's a heterogeneity there, and really optimizing who to target, what to target, is going to be important as this field moves forward. So just some conclusions. Increased exposure to environmental microorganisms is associated with decrease in childhood asthma. Appropriate early development of the microbiome is associated with risk of asthma development. Alterations in the respiratory microbiome have been associated with specific asthma endotypes and exacerbation in pediatric asthma. And then there's differences in the intestinal metabolome. And again, what I mentioned with the mechanistic studies. So these are all my references. I appreciate you all bearing with me as I too was learning about this topic. And the word of the day is microbiome. I appreciate any thoughts or questions or insight others might have who have more experience or expertise.
I have a question, might be a little bit illogical, but if certain organisms are known to be beneficial, a lot of this work is looking at abnormalities of the microbiome that might provoke asthma. But if certain organisms were uniquely helpful, has anybody thought about like intentionally colonizing the nose with that microorganism to be preventative or protective?
I haven't seen, I think that's a fair question. I haven't seen that done. A lot of the interventions were done via oral and changes and things there, like probiotics and
Right.
prebiotics in the intestinal tract, but not the nasal cavity.
Other comments, questions?
This is Vinod. This is Vinod
Yeah.
here. Can you hear me?
Go ahead.
Yeah.
Yeah, thanks for a wonderful talk. Maybe I just just following up on Len's question. I know there was the for the food allergy study, the gut microbiota replacement study. I'm not sure where that went, but I think one of the things you alluded to was the connection between gut and lung. So has that been looked at? Where in the ones there was a gut microbiome replacement, or the studies that they were giving certain microbiomes, how that affected asthma development in those studies? Are you familiar with that? Thank you.
Sorry, can you repeat the last part of your question? And cut out a bit.
Oh, sorry. So I know there was, I'm not entirely followed up on those studies where gut microbiome treatment for food allergy I don't know, certainly nothing that he approved has come up at this time, but they were talk about, giving capsules of certain microbiomes, to, in terms of atrophy and developing the gut microbiome diversity. I don't know where, I don't know if that's been looked at in the context of how those, cohorts did with regards to asthma development. I think you did allude to the part where gut microbiome and asthma, there's a, lung, there's a connection. So even though you may not want to put something in the nose, we're treating something in the gut could have positive benefits on the asthma development.
Yeah, I think that's certainly possible. I don't know that it's necessarily going to directly change, the composition of the microbes at one site versus the other, but I think that certainly the immunologic effects can be impactful in by similar with food allergy and changing the interactions in the gut by giving certain microbes. I think that at least through these studies and a lot of the the basic and translational work, that certain microorganisms can promote differences in the immune system that can contribute to asthma and the lung, immunity in that regard. But I don't know if in those studies for the food allergy, whether or not that impacted risk of asthma. So I'll certainly look into that. That would be interesting to see.
There's a comment in the chat, basically just supportive of the work in Germany, if I understand the comment correctly.
Mm-hmm.
Yeah, what it doesn't address, though, and probably no one has studied it, is if what you're exposed to on a farm becomes part of your microbiome, or if the mechanism of protection is totally different, like the hygiene hypothesis.
Mm-hmm. Yeah.
Well, certainly not an easy topic, not one where there are definitive answers. Any other comments from our audience?
Thank you all. I appreciate you listening in today.
Yeah, well, thanks for reviewing a complex topic.
All right. Take care, everyone.
Okay, well, we don't have any other comments. We'll end a little bit early. Thank you.
Thanks.